So today I want to share a talk with you that I recently did at the annual meeting of the American Society of Agronomy, which was in Baltimore, Maryland. I was asked by the society to give a talk on serendipity in science, how that occurs, accidental discoveries in science and how they impact our science. And of course, my immediate thought when they asked me to give a talk about serendipity in science is, so would you think my whole career was just based on luck? That's why I'm here talking. But they said, no, we don't think that. But you've had some significant discoveries, and we'd like you to talk about how those came about. So I decided to title my talk, Chance Favors the Prepared Mind. And I've always loved this quote. I first read it when I was in graduate school, and I thought I want to dedicate my career to having a prepared mind so when those chances come along, they're a big deal. So this seems to make pretty intuitive sense, but what's a prepared mind really mean? You know, an analogy that I thought of is, what if we said, chance favors the prepared athlete? Well, that would make perfect sense. Athletes spend a long time preparing their minds and bodies for that one catch in some great game. But that wasn't luck. That was a lot of skill honed by years of practice. The same principles relate to this with scientists. The prepared mind means learning all the principles of your discipline, the equations that make up your discipline, the theory, the models, the literature in your discipline. It means really becoming a true expert in your discipline. That's what prepared mind means. And then when you come along and see the odd things, you're prepared to say, that could be something right there, and you follow it up. Lesser people might say, now that was bad data, just forget it and move on. So this quote came from Louis Pasteur. He lived in the 1800s. He was a great philanthropist. Everybody knows about him. The germ theory of disease came from Pasteur. He was always working with sterile systems. It's amazing stories of his life. But when he wrote about this, he got the Nobel Prize. When he wrote about this, he didn't have 5 succinct words to describe this principle. Here's what he wrote. Well, even before I get to what he wrote, let's take a look at what a prepared mind might be. Here's some data, right? So this is an X plot, an input variable. Here's Y, a plant response. You get data like this. Now, we would normally say the data's like that. This is a common principle for a lot of things. So the more you give X, this is nonlinear. You make measurements, you get this point right here. The first inclination is, that must be bad, just throw it out. But you measured it. What happened with that? Let's see if I can get back to this. You measure it, just throw it out. We have statistical principles that say this is so far off the line, just forget about it. You can throw it out. But there's an example of something that might be real. Let's take this out here for a second, that little line right here. And we're going to go back to this. Now, some people might take this and they have a high-speed computer and they'll attempt to fit this with a 10th order polynomial that looks like that. And now you got to come up with the principle that says right here there's a big inhibitory effect of X on Y. So just that simple data set alone gives you a lot of examples of the challenges we go through when we make good measurements, very careful measurements, and we're sorting good data from bad data and looking for those anomalies that lead to great breakthroughs. Pasteur didn't say these 5 concise words. Here's what he originally wrote for his Nobel Prize. In fields of observation, chance favors the prepared mind. But look at this. This is true in all fields, not just in observation. So we can edit that out. Chance favors only the prepared mind. And not only that, we don't need the word only. We edit that out too. Now we get to his quote. So Pasteur, like a lot of people, had good editors as stuff to make the concise statements. And that's part of science, the conciseness that leads to new breakthroughs. So we often think scientists must say when they have a breakthrough, they say, eureka, I found it. But that's really not how science works. You rarely get to something you expected. The most common phrase is, that's funny. And that's the part that leads to these great breakthroughs. This came from Isaac Asimov, this quote. And here's a book that I've had on my shelf for years, Serendipity, Accidental Discoveries in Science. And this is loaded with examples of unhappy accidents that occurred in science. It's an interesting book. It's still in print. So let's take a deeper look at this. In 1963, Science magazine had an editorial, and the conclusion of the editorial was, serendipity is not a substitute for hard work. And that's a gold standard in this. They were being criticized. You should just fund the scientists that are lucky. Yeah, there's not lucky scientists. There's ones that have hard work and they have prepared minds. So they were pushing back on this. There's an old phrase, the harder I work, the luckier I get. Yeah, great. But look, hard work is in here. Work is in here. Work is something you don't like doing. And in order to do a lot of it and feel good about it and be creative, you got to do the stuff you like doing. You have to like what you do. So I always say, this isn't work. This is something I enjoy. And work is something I don't enjoy. That's one of the keys to doing a lot of something and then being able to pick out the happy accidents that occur. So let's get rid of this. Here's another big thing about statistics. Statistics is not going to lead us to great breakthroughs. It helps us determine mathematical probabilities, but I love this quote. If your experiment needs statistics, you ought to have done a better experiment. This came from Ernest Rutherford, Nobel Prize in Chemistry. You can believe he wanted clean experiments where he didn't need a lot of reps and he didn't need a lot of statistics to sort out his results. And certainly in our research here, we try to do simple, clean experiments so we don't need a lot of replicates. So the next section of this talk is about this intersection between theory and measurement. And we get to a point right there. Knowledge is really the extent to which theory agrees with measurements. Now, theory is another term for models, and models are equations. And if models predict something and that's what we measure, we get confident that we have it right. This is in this room up here on a board by our clock to keep people thinking about this. And really, this quote in here says, never believe a measurement unless it can be substantiated with theory. And conversely, the degree of confidence we can have is the degree to which these two things agree. So theory is what you learn in school, theory is what prepares your mind, and measurement's what you do in the lab, and you look for when they converge. Now, to pick out these serendipitous measurements, we need wisdom. So it's a three-way interaction between all three of those things that gets us there. So here's another quote that's just striking. Everybody believes a measurement except the person that made it. Now, the person that made it knows how hard it was to make that measurement, and they're not sure they got it right, but everybody else thinks, oh, no, it's measured, it's fine, it's the gold standard. The corollary to this is Nobody believes a model except the person who made the model. And the modelers are always saying, I have a model, look, it's right. And people say, no. So the bottom line of this, we should believe measurements less, and we should believe models more. They put equal weight on both of these to guide us. Now, the place where I think much of this came from is Back to the Future, where they're making a lot of measurements to go into the future, and they make a lot of models back and forth. So it's a nice corollary to this quote. So, let's see, we'd get this off the slide. The analogy I have used to emphasize this link between theory and measurement is getting to the top of a house with 2 ladders, and the rules of science say you can only have 1 foot on each ladder. And 1 ladder is measurement, 1 ladder is theory, and you get to the top of the house fastest when you put 1 foot on 1 rung and back and forth and back and forth, and up you go on the ladder. If you tried to do only measurements, you maybe could get there hopping on 1 foot on the ladder, but it'd be awfully hard. And only theory, hopping on 1 foot would be awfully hard. Now, another part of this is if you want to be really safe, you should tie your ladders together so that the whole thing is more safe. And that's good, because we want to link theory and measurement intimately back and forth. Here's some examples of that. Here's theory, there's measurement. Let's take these off of here. Back and forth, and here we go. In, up, out. But boy, they're far apart, and this takes a while to get up there. What if we more intimately link them? Now we have faster progress because they're closer together, back and forth, and then finally they become one and the same in the same person. Theory, measurement, evaluate, that makes for excellent progress. I want to further emphasize this with some pictures. Here's theory. Albert Einstein, all he ever needed to do research was a box of chalk. And it's wonderful. I'd like to be his department head. I'll be needing another box of chalk this year for my research budget. Here's our theory. We do all kinds of empirical replicates in the field that look like this. Theory and measurement. Excellence emerges from continuous coupling. We move these together, it's continuously coupled, and we make faster progress. We sort out the bad data from the good, and we improve our models by doing this. So often in research, we get this out of balance, and we do a lot of empirical measurements, and we have very inadequate theory to explain them. In some labs, the reverse is true, and we have a lot of theory, but not very much data. This often happens in climate science, where we have a lot of models. We rely heavily on models and just a little bit of data. But in both of these cases, it's out of balance, and it doesn't lead to the fastest progress in science. Now let's switch to a little bit of data that we have here in our lab. This is a graph of the colors of light at night and how they affect flowering in cannabis. So we always say a green light is safe. If you want to work at night, always use green light because that has no effect on plants. Don't use red or blue. This is days after start and flower diameter. So the steeper the line, the faster the plants flowered. So if they were completely dark, they flowered fast. If we gave them blue light, and I can show that in blue on here, they flowered equally fast. So in this study, blue light was safe, no effect. Look at red light. Red light's the opposite of green. Flowering was inhibited by red light right here. And the flower diameters were smaller because the plants perceived red light at night. Here's the point, look at the green. Green, in this study, was just effective as red. So it's not the case that green light is a safe light. Plants perceive the green light. How can that be? If plants reflect green light, how can that be so potent? That's why we think it's safe, because they just reflect it. I'm going to put up here a diagram of how well light penetrates plant leaves. Green light is reflected, but that also means it penetrates deeper into leaves. This is a diagram of the cross-section of a leaf. Here's the top of the leaf. Here's the bottom of the leaf. And this distance is just like 1 millimeter. It's way blown up. Blue and green are absorbed very quickly in the surface layer. The green photons hit and they bounce around and they penetrate the leaf. And this means the green photons get to the site of perception of the light, which in this case is the phytochrome molecule. And even though those photons don't have such a big effect, they're not zero, and they're getting to the site of perception. So this only came from our understanding of principles. We said, well, this can't be correct. Just throw the green light data out. No, it led to a new principle, and here's the paper that resulted from it, improving the predictive value of phytochrome photoequilibrium in a consideration of spectral distortion in a leaf. So this data led to this paper. Now, having said this, green light is still the best light to use because our eyes are really sensitive to green light. So use green light if you have to work at night, but try to use a minimal amount of green light. It is not completely safe. Let's take another example. This is weighing lysimeters in our greenhouse. Go back to this. These are load cells. We weigh plants. We measure transpiration with this. Here's a growth chamber, and this is cotton on these and corn on these chambers. So we can make very accurate measurements of how open the stomates are and how much transpiration there is. Let's look at the data. This was day and night, 2 days, transpiration rate, which is right here in purple. Now that is, and let's see if I can show this in purple, right here. It's low at night, not zero. Here's zero right there. High in the day, low at night, high in the day. This is our measurements, just like that. Stomatal conductance, that's how open the stomates are. That's shown in green. The stomates open in the day and close at night, but look, they're not completely closed at night, and this number right here is about 0.4 open and wide open, and the day is about 0.9. So in this data, the stomates are about half as open at night as they are in the day. The reason this is higher than this is it's very humid at night, and the leaves are cooler, so that transpiration should be much less because there's not much of a driving gradient. Conductance is the ratio of driving gradient to transpiration. But this is remarkable. Now, this data was collected some years ago in cotton, but we know this also happens in cannabis, which is a very big deal because we have a lot of flux of water at night when the stomates are supposed to be closed, and they're open. We have to run dehumidifiers or we get plant disease. All the textbooks say stomates close at night. And that sentence in textbooks is just wrong. Except if we give the plants some water stress, a minor water stress, they quickly start to close their stomates at night. In this same data, had we stressed these plants a little bit with water, now the conductance would be down here. It looks like this. So if plants, like many of our cannabis, if they're grown with no water stress, they don't close their stomates at night. It's a big, big deal. Look at this curve. This was in electric lights. Let's get rid of these lines here. this is in electric lights, and I'm going to have to do that. There we go. The lights in this were a square wave. They came on and looked like that. This is not sunlight, it's an electric light. All of our principles say that transpiration rate should follow the light. It should go up and high and come down, and instead it's a curve in here. And it happened every day. It should have been a square. It's a curve in here. Our models don't predict that. The measurements in here didn't match the models. When we got this data, we very quickly went to multiple plants. Let's go right here and looked at this. Here's a square wave of light. All plants do this. In the afternoon, it's like they get tired and they this is transpiration rate, or this is stomatal conductance. They start to close their stomates in the afternoon. And they don't even open them till about 11 o'clock in the morning. It's just remarkable that plants do this. And it took controlled environments to be able to study this. This has, now look at this. These lines are close to zero down here because the plants got a little bit of water stress. This has big implications for designing air conditioning systems and for humidity and disease control. These stomates are wide open in the middle 1/3 of the day, but it takes them a few hours to get there and they close their stomates for a few hours in the afternoon. Now let's go back to another fascinating thing about this. This is a well-watered cotton plant and two water stress cotton plants. This same thing happens in cannabis. Here's the opening. Took a long time to open, and then they started to gradually close. There's night right there. But look at these drought stress plants. They also take a while to open. They stay open, maybe about half. This is a measurement of how drought stressed they are. But look what happens here. They go nuts. There's noise. That is not noise. The whole plant is cycling for water. It opens its stomates. Oh, we overshot, and it closed them, and open, closed, open, and closed. We've seen this repeatedly in plants. This is anomalous data. This is the kind of data you go, that's funny. And then you find out you can repeat it. This has big implications for design of controlled environments where we spend a lot of money taking the humidity out of the air. Let's get rid of these, and we'll do, those lines are off. Now let's move to another huge area for us here in the crop physiology lab and my colleagues. This is far red photons. You can just barely see far red right here. We call these the forgotten photons because they're on the edge of our vision. We can barely see them. We've done a lot of work with far red photons and their photosynthetic value. This work was done with my long-term colleague, Mark Van Eersel at the University of Georgia. He was here last summer with us, recorded videos with us. He's just been a joy to work with. He's now got an endowed chair position at the University of Georgia. And Shu-Young Zhen, who is a very talented scientist, she got her PhD with Mark, then she came to Utah to work with me for a postdoc. She is now an assistant professor at Texas A&M University. Together, the three of us have published six, 7 papers on this, and I want to review this evidence because it has the potential to be a profound implications for all of plant biology. Here's lettuce, no far red, and 10% of the photons is far red. Now, you don't have to be a rocket scientist to see those far red photons. They're doing amazing things to this plant. Some of this is due to what we call a shade avoidance response. They think they're in shade, so they just spread out. They spread their leaves bigger. That's not surprising. We've known that forever. But with careful measurements of photosynthesis, we have shown that some of this profound effect is also due to far red photons. That is a big deal because for years and years we've said far-red photons do not cause photosynthesis. So it's an amazing claim to say they do. When we have amazing claims, we should always fall back on this mantra. Extraordinary claims require extraordinary proof. Now, this was popularized by Carl Sagan in Cosmos, but this is an old saying. So now what I want to do is show you the extraordinary proof for far-red and photosynthesis. This is it's a very concise summary, but this started in 2017 with Journal of Plant Physiology. Mark and Shu Young in Georgia studied the light reactions of photosynthesis and very clearly showed these far-red photons had a beneficial effect. They followed it up with laser diodes a year later, published another paper, Physiologica Plantarum, showing that the wavelengths of far-red up to about 730 nanometers were clearly helpful in the light reactions. Then, after that, Shu Young came here to our lab in Utah, where we have a whole series of whole plant gas exchange chambers, so we can measure photosynthesis on whole canopies. Shu Young and I published a paper in 2020 in Plant Cell and Environment, one of our top journals. Far-red photons have equivalent efficiency to traditional photosynthetic photons. Implications for redefining PAR. PAR is photosynthetically active radiation. We kept working on this in longer-term whole plant studies, published a paper on far-red photons in Frontiers in Plant Science, so more accumulating evidence, more papers, and now our colleagues are getting into this, and there's other papers from other laboratories showing far-red photons have photosynthetic value. Note that all these papers are pretty recent papers. Now there's one thing that's critical here, and that's the mechanism for this. Here's my diagram of how the light reactions of photosynthesis work. The Stark-Einstein law says one photon excites one electron. This is how solar panels work. This is how photosynthesis works. A photon comes in, excites this guy, this electron gets excited, and then it's passed down this mill, this turns, it makes ATP, makes energy, another photon hits this guy, boom, another electron's excited. So it's a two-stage process. Photosystem 2 comes first and photosystem 1 comes second. This is because we discovered this first, and then we later found out there's two. Water gets split right here to oxygen, but here's the key. We have known for a long time that far-red photons excite this photoreceptor in photosystem 1, and that is called the Emerson enhancement effect. It's been known for 50 years, but it's been little studied until now. When we got far-red LEDs, we can do more rigorous studies of this. You can pause this and look at this diagram, but this is electron transport in photosynthesis, but simplified to show electrons getting excited. It takes four photons here and 4 photons here, either classic or far-red, and it takes 8 photons to get through the whole thing and fix CO2. So we have extended this now to plants in sunlight, not just electric light, sunlight, and very recently, July 2022, we published a paper, Photosynthesis in Sun and Shade, Surprising Importance of Far-Red. This extends the work to sunlight, and this is in New Phytologist, is one of our top journals. We hope that this will help people understand the value of far-red light, which is a very big deal in the world of cannabis because far-red photons are very efficient. They're long wavelength and they're very photosynthetically efficient. Here's the classic McCree curve for photosynthetic efficiency. Our definitions, it cuts it off here. These photons are not very many, and this curve is pretty accurate. They drop pretty fast. Over here, we got 700. There's the classic definition. Now we add this recent work. This curve has a bulge over here. So it looks like this, and it goes to 750. The old definition is called PAR, photosynthetically active radiation. The new definition is called EPAR, extended photosynthetically active radiation. Apogee makes a sensor to measure EPAR, and it's a better predictor of photosynthesis than the old PAR. So on a close here. This is a diagram done by a Utah artist, James Christensen, and I just love it. This is what we do in science to get serendipitous discoveries. We take our equipment out, and sometimes our labs look like a bad day in a spaghetti factory around here. We take it out for a drive and test it and see what happens. And if you want to know how to get serendipity in science, build equipment, homemade, doesn't cost much, and take it out for a drive and see what happens and be prepared for anomalous findings. Thanks for listening. I hope this inspires you to go on and make a lot of measurements in science.